Okay, good afternoon, everyone. Good afternoon, everyone. I think we can start. It's 12:02, 2 minutes past 12. The faster we start, the faster you get to go downstairs and do some really cool experiments. I like the experiments in this lab. This is just me. It's one of my favorites, and I hope you enjoy it as much as I do. So today we're going to talk about a very important group of proteins, and these are enzymes. You're going to learn more and more about enzymes as you progress in your medical curriculum. You will take that in detail, for example, on the second semester in the biochemistry course. You're going to talk all about enzymes, and one of the things that you're going to learn about enzymes is that there are factors that increase their activities or that affect their activity and how. First of all, can you tell me, what do enzymes do? Speed up reactions. Excellent. So they're not really involved in the breaking down or the building of things, but they facilitate it to happen, basically. So, and you'll talk in more detail about exactly how does it facilitate reactions, but this is beyond the scope of the lab. But what you do need to know is that their function is mainly as a catalyst. They do speed up reactions, they make the reactions faster, and basically there are factors that affect how well they can do their job. Can you guess what would affect those catalysts, those things, those proteins that speed up, they're supposed to speed up reactions? Can you think of factors? Temperature, excellent. So the temperature. Enzymes usually can only work in a certain temperature, and it would make sense that most of them will prefer temperature close to the human core temperature, so somewhere around 37 degrees Celsius. Excellent. Yes? pH. MashaAllah alaikum. So pH. Every enzyme will have a pH that it likes to work in. Can you give me an example of an enzyme that only works in an acidic? Catalase? I don't know about catalase. No, an enzyme that works in an acidic environment. Pepsine. Okay, so pepsin. All right. And then you have other enzymes that prefer to work in a more alkaline, and then more enzymes that prefer more neutral pH. So that definitely the pH is a major contributor to enzyme activity, and this differs between enzymes. Some like acidic, some like more neutral, some slightly alkaline. And that is basically the purpose of this lab. I'll talk about these factors that regulate enzyme activity, and when you go down, what you're going to do is you're going to do different experiments that will prove that yes, enzymes need an optimum temperature. Yes, they do need an optimum pH, and you'll see with your eyes how the activity of the enzyme will change based on the conditions. All right, so these are the objectives. You are expected to carry out 5 experiments related to enzyme properties, and that includes specificity. So enzymes are specific. You don't have an enzyme that can work on all reactions. Our body is way too complex than that. So every reaction has a set of enzymes, and they're only specific for that given reaction. So you will do an experiment on that. You'll test the effective temperature as a contributor that regulates or influences enzyme activity, pH. And the other thing that we did not mention is how much substrate there is and how much enzyme there is. So if you have a lot of enzyme but not enough substrate, then the enzyme's not really going to do a lot of activity because there's hardly any substrate. And vice versa, if you have a lot of substrate but you don't have a lot of enzymes, you can imagine that there won't be as much activity compared to if you have high concentration of substrate and high concentration of enzymes. So the effect of substrate concentration, the effect of enzyme concentration, that also influences and determines how much activity you would expect from that enzyme. And for your experiments, we will be using catalase, as you mentioned. So it's good that you know catalase. And catalase needs the substrate for a catalase. And I'll say basically substrate is the terminology we use for that thing that the enzyme acts on. So the thing that the enzyme acts on is known as a substrate. And because enzymes are specific, so catalase specifically acts on hydrogen peroxide, or H2O2. That is hydrogen peroxide. So it acts on it, it binds to it, and it facilitates the breakdown of hydrogen peroxidase into oxygen and water. So you will do these 5 experiments. You will change things up and see how does that translate in terms of, once you change these things, how does that change the activity of the enzyme? Just some terminologies. Remember, because enzymes are required to facilitate, they're there as catalysts, that they facilitate chemical reactions. I need you to familiarize yourself with basically a term that we use that refers to chemical reactions in the body, and that is metabolism. So metabolism is the general term that we use to refer to all the chemical reactions that happen within our cells, within our body. And those chemical reactions, they either build things or sometimes they break things up. And those that contribute to the building of things, it could be building of protein, building of muscles. This is referred to as anabolic metabolic reactions. And the opposite is for breaking down things, and that is, those reactions are typically referred to as catabolic type of reactions. So in very simple terms, this could basically depict what an anabolic type of reaction would be. This could be 2 amino acids that will give this product. So if this is your initial reactants, if this is your substrate, you'll end up with one product because you're building something from other things, right? So this is an anabolic type of reaction. But sometimes, and I apologize that this is smoothed, sometimes you can have one substrate that is broken down to other 2 smaller things. So this is breaking down, so it is a catabolic type of reaction. And what you're going to do is kind of similar to this. So you have your substrate is the H2O2, hydrogen peroxide. In the presence of the enzyme, it'll be broken down to water and oxygen. And water and oxygen, you will see it with your eyes. How? As bubbles. When you see bubbles, you know that the reaction is taking place. All right? And, all right, that's, again, anabolism. You need energy to build things. Catabolism, you typically you break things and you release energy. And the way the enzymes act, all right, this is catabolism. So you have one substrate that will be broken down into smaller substrate products, and with that you release energy. All right, so if this is, for example, your enzyme, and this is the substrate, this enzyme will help this substrate break up into 2 different components. And then it will release it. So enzymes, they are not used up in the reaction. They're just there, they facilitate it, and then they leave as it is. So it'll remain the same way. It's not going to change. So they are not utilized in the reaction. And there's a reason why we put it. If you notice, that also brings up the issue of specificity. Usually, these enzymes can bind to specific locations, a location called the active site on the enzyme. And that active site is kind of like the lock, and your enzyme is your key, or vice versa. Your enzyme is the lock, has an active site that is the lock, and your substrate is the key. And then the issue of specificity, just like the key can only open one door, usually enzymes are specific. Only one substrate will be acted upon by one enzyme. So let's see, how does an enzyme work? Enzymes are a biological catalyst. They speed up chemical reactions in our body without them being consumed in these chemical reactions. And these reactions could be anabolic, it means they build things, or they could be catabolic, in which it means that they're breaking down things. What is the substrate? It is the substance which the enzyme acts upon, and the active site is a pocket-like structure on the enzyme surface where the substrate is going to bind. I'm going to change this slide. I'm sorry, this is kind of overlapping with the text. But yes, so these are essentially some terms that you need to know. So if this is your enzyme, which is your catalyst, then that enzyme will have something unique shape, a unique part of it, a unique structure known as the active site. On that active site, you will have a specific substrate that it'll bind to. And once it binds to it, it'll create something called enzyme substrate complex. Okay, so it's a complex composed of enzyme and substrate, so it makes sense that we call it enzyme substrate complex. And then that enzyme will facilitate the breakdown of this substrate into two products and then releasing them. So this is essentially the steps of enzyme actions. These are the terminology that you need to know. What is an enzyme? What is a substrate? What is an active site? What is an enzyme substrate complex? For this, it's enough for me to know this for today's lab and the factors that influence enzyme activities, but for sure you're going to have much more detailed lectures on enzymes in the second semester of year 1. All right, so enzymes are known to be specific, and they need an optimal temperature, they're affected by pH, so they need an optimal pH, and they are also affected by the substrate and enzyme concentration. So the more you have substrate and enzyme concentration, the more reaction that will happen. All right, if you don't have enough enzymes, no matter how much substrate you have, it will be relatively slow. Or that would kind of affect the activity of the enzyme. So you're going to do 5 experiments because you're going to look at those 5 factors that affect enzyme activity. And as I said, in your experiments, you're going to use catalase as an enzyme and hydrogen peroxide as the substrate. And of course, catalase is found in our body. It is a natural antioxidant enzyme. It's produced in most organisms, and the main action is to that enzyme catalase to basically break down any toxins, any hydrogen peroxidase that could be present in the cell into less harmful molecules or substances such as oxygen and water. We don't want H2O2 in our cells because this is an oxidizing agent. It can damage the cell, it can create mutations, it can turn cells into unhealthy cells. But naturally, we have a defense mechanism against hydrogen peroxide, against this oxidizing agent. And that enzyme is catalase. So it's very important to protect the cells. So this is experiment number 1. You're going to have 3 tubes with 3 different substrates. One will have water, one will have sucrose, one will have hydrogen peroxide. Once you have those, in each of those 3 tubes, you're going to add on top the catalase. And then observe for a reaction. Are you going to see the bubbles? It means, did you break down hydrogen peroxide? Or basically, did something happen? In this case, of course, because catalase is specific for H2O2, here there will be a reaction and you'll see bubbles. So this addresses the first item, which is the issue of specificity, enzyme specificity. Mafhuma so far? All right. And now, so this is basically something similar to what you see. You could see nothing here in tube 1, tube 2, but then you would see these bubbles. So how do if I ask you, how do you measure the reaction and whether a reaction took place? You'll say, oh, by the appearance of bubbles. And then you will do something similar. You'll take 3 tubes, and you can have basically catalase enzyme. That is what we're going to study. We're going to take catalase as an example of an enzyme, and place each tube in a different temperature. One, you can put it in a hot water bath around 100 degrees, one around 37 degrees, and the last one, you can put it on ice. So you have 3 different temperatures. You put your enzymes, let it sit in there for a few moments, then add on top of it the substrate, which is H2O2, and see where do you see reaction, and where is the reaction and the amount of bubbles, where is it more? Is it more at 0 degrees or close to 0 when you incubate and you leave the enzyme on ice? Or is it higher when you leave the enzyme in maybe 100 degrees Celsius? Or is it highest when the temperature of the enzyme in the tube is closest to the core temperature? What do you think? You'll let us know, okay? So this is essentially what you do. Just make sure that it's at the right temperature. You can use ice, 100 degrees, and you can use 37 degrees. Put your tube that contains your enzyme, and then on top of it, add your substrate and observe the reaction, okay? So that is the second thing that I would like you to know, and you will come to know that the conclusion you should draw is that, yes, the rate of the reaction depends on the temperature, because if the temperature is too low, somewhere around 30, then the rate of the reaction will go way down. If it's so high, anything above 50, then the rate of the reaction drops. So there's always that window where you would expect the enzymes to act the best, all right? And luckily, our temperature is somewhere around here, and therefore, it is still within the range for optimal temperature for enzymes. All right, so then this is the result that you would see related to optimum temperature, and these are the bubbles. So measure them and measure their height. See if there are differences in height, which could indicate differences in activity. All right, especially if you're adding the same amount of substrate and enzyme. So that difference in change of the reaction can be traced back to the difference in temperature. The third thing is about enzyme action, concentration. So you have 3 tubes with different concentration of catalase enzyme, different amount really, to be more precise. And then I'm going to add the same amount of substrate on top. I'm not changing the amount of substrate, it's the same. Let's say I'm going to add maybe 500 microliters, let's say, in each tube. So the substrate is the same, but the amount of enzyme is different. So I need you to do this and see how does the enzyme concentration affect how much bubbles you will see. All right? And I want you to do the opposite. Change the substrate amount and add the same amount of enzyme, and see how would that affect the enzyme activity and the bubbles produced. I want you to come up with these conclusions. So typically, as you increase substrate concentration, you would expect there to be higher rate of reaction. Same thing with enzyme concentration. But also bear in mind that it's very easy to plateau, reach a flat line. So this is only effective for some time, but eventually, for example, you could add a lot of substrate, but if all the active sites are saturated in the enzyme, it doesn't matter how much substrate you're going to add. It reached maximum velocity, that reaction. So that is not always linear. It's not علاقة طردية بشكل تام. It does plateau. But it is a positive correlation with enzyme or rate of reaction. So substrate concentration and enzyme concentrations will positively increase rate of reaction to a certain degree. So we mentioned the specificity experiment, the temperature, and then we had the substrate concentration, the enzyme concentration. This leaves the last experiment, the effect of pH. So you'll have catalase, same amount, but you'll add a solution on top. One that is acidic, one that is neutral, one that is more alkaline. And then you'll add the same amount of substrate, your H2O2. So I want you to go and I want you to tell me, is does catalase like an acidic environment, an alkaline, or does it like a neutral environment? So this is the final experiment for you. And it's natural that this is different for every enzyme. Actually, every enzyme has a unique optimum temperature. So for pepsin, it's something around 2, very highly acidic. Amylase or salivary amylase is like close to 7. And trypsin is more alkaline, around 9 optimum pH. So tell me, what is the optimum pH of catalase? So please, I encourage you, because today I encourage you basically to go as soon as possible downstairs to get the most out of today's lab and enjoy. Please try to come up with conclusions and explanations to your findings. Thank you so much.